ramp up in issues of convalescent plasma, dexamethasone. What are those therapies that we can use before the vaccine is ready? And for those of you who joined the White House conference last week, for 2 hours, we had Donald Trump, we had Deborah Birx, we had Francis Collins, Steve Hahn, Dr. Fauci, Jerome Adams, the Surgeon General, all speaking about convalescent plasma, safety, efficacy, where we're going with FDA, EUAs, and that sort of thing. So lots of progress in the communication. We saw last evening 2 articles, the New York Times and the Wall Street Journal, both creating eloquent summaries of the journey of where we are in convalescent plasma, the EAP program, and the next step of randomized controlled trials. We'll also be seeing in the upcoming days a lot of discussion on back to school. And what are we doing to get back to school safely? And what are the social distancing and mask rules? And what are we going to be able to do with remote versus in-person learning by county, given the situational awareness of each geography? So we're very, very heavy into the pre-vaccine communications and activities. But today's discussion, of course, is anticipation as we get to the post-vaccine era. What are the vaccine approaches that are being tried? Are they humoral immunity and cellular immunity? Are they likely to work? Are they going to be persistent? Are we going to need multiple doses? So many questions and unknowns. And of course, we worry that herd immunity is going to be a little challenging, given there may be some resistance to these emerging vaccines that are being developed at a record pace. So today's discussion, very, very important to plan for the next step. And I'll turn it back to you, Brian, and look forward to the dialogue.
Great, thanks, John. So let's go ahead and dive right in. I'll introduce Dr. Lauren Quattrochi. Dr. Lauren Quattrochi is going to walk us through some of the fundamentals of the different vaccine types under investigation, their anticipated advantages, disadvantages, as well as the status of the ongoing vaccine trials. Dr. Quattrochi is the principal biotechnologist here at MITRE. And so we have with her, alongside her, Dr. Michael Berry and Dr. A.J. Venkatakrishnan from Inference. Dr. Berry's from Mayo Clinic. I'll let John introduce them as they take their turn to speak. But so with that, Lauren, would you like to provide a few opening remarks? You might be on mute, Lauren.
Yep, you've got it. Okay, so hello everyone, and thank you for taking the time today to learn a little bit more about the vaccine pipeline and what we have in store for you. So I guess, Susan, let's kick it off and go ahead and share some slides. Fantastic. So to give you guys a quick overview about my background, why I'm here, I've had the pleasure of working with multiple agencies within the therapeutic, biopharma, and vaccine space. That includes clinicaltrials.gov, the Assistant Secretary for Preparedness and Response. In that one, we were looking at the OWS, or Operation Warp Speed, vaccine manufacturing feasibility, as well as creative and disruptive vaccine facilities that we could use as an alternative if we need to ramp up even more beyond the 300 million vaccines that Operation Warp Speed is planning. I've also had the pleasure and am currently working with JPEO on the DoD side, leading and spearheading 3 different avenues for them, one of which is their COVID DNA vaccine with Inovio. Another is a USAMRIID hantavirus vaccine, and the third being monoclonal antibodies against plague. So I've had other opportunities as well here within MITRE, including the National Institute for Allergy and Infectious Disease, and spearheading vaccine acceleration programs there for high priority pathogens that we know to be potentially relevant as emerging infectious diseases. So with that, I'll go ahead and give a little bit of background. Before MITRE, I was at Pfizer where I worked in 2 different divisions, the first of which was research formulations, where you look at the physiochemical properties of the drug and decide how best to administer it so that you get the pharmacokinetic profile you need. And then the other was the ADME portion of things, so the pharmacodynamics and how not just what the body does or the drug does to the body, but what the body does to the drug. And so with that in perspective, let's go ahead and jump to the next slide. So the whole reason that we are here is that the US is performing pretty poorly in terms of COVID-19. We are winning, and it's a game that we don't want to win. Right now there's 18.5 million confirmed cases. Those are just the ones that have been confirmed. We know for a fact that this is probably higher than that. A recent CDC study that came out said that it could be that we are undercounting by about 10-fold. As far as confirmed deaths, we are over 700,000 worldwide. And if you look at the US case count, you'll see that we have a death rate of about 3.2%. And this was pulled just today. So moving on to the next slide. All right, so the big take-home message is that the vaccines are coming. These companies have been doing incredible feats to get these through the door. A lot of these have been combining clinical trial phases to expedite what they need and the mile markers that they need to achieve to prove both efficacy and safety. However, safety is going to be the #1 top concern, both of FDA and of any of the companies, even beyond that of efficacy. What we imagine to be is that the first COVID-19 vaccine that hits the market likely won't be effective enough to quench the entire global pandemic. It's going to take an entire village. And I imagine that the 300 million vaccines that are planned for Operation Warp Speed and for the US general public won't be one type of vaccine. It's going to be a compendium of different types of vaccines. So we're going to see then post-market, after they hit for the general public, how those vaccines really behave in the real world. So the COVID-19 vaccines, they may need to be periodically re-administered. And so it's going to be important for anyone in the public to remember which vaccine they actually get. This could be very similar to our annual flu vaccine, where we only have a 20 or 60% effectiveness within the population. And so we have to revisit that in getting those vaccines to maintain that herd immunity if and when we can achieve it. At least within here in the US, the vulnerable populations will be prioritized, and there are different ongoing discussions right now around how we're going to prioritize that. The national academies as well as ACIP have been looking into how we are going to prioritize the vaccine distribution. We've clearly identified that, you know, things like nursing home and frontline workers, these are going to be areas where we do need to prioritize those individuals. Right now, if you check the World Health Organization's website, I checked today, and you'll see that there are 26 vaccines currently in clinical trials on the global stage and 139 vaccines in preclinical development globally. And again, all 26 of those vaccines are going to come to market, but it doesn't necessarily mean that they're going to be the best vaccines. And so it may be a bit of a waiting game so that we can see how they behave in the real world. Go ahead and hit the next slide. Okay, so for those of you who may not be familiar, there is Operation Warp Speed, and I wanted to very briefly go over this if you haven't been tuning into the news quite so recently. So the goal of the Operation Warp Speed, it's a conglomerate of both HHS and DoD efforts, and the aim is to deliver 300 million doses of a safe and effective vaccine for COVID-19 by January 2021. It looks as though some of those vaccines may be ready at that time. However, it will probably be, again, for those vulnerable populations. Distribution for maybe the general public or folks like me may take a little bit of time. So I would expect that it's probably another 6 months or so until it actually becomes a widespread vaccine. So as well as vaccines, they're also looking at therapeutics and diagnostics. As far as the leads, well, HHS Secretary Alex Azar and then the Defense Secretary Mark Esper oversee OWS, but kind of the nuts and bolts are run by Dr. Slawi as the designated chief advisor, and then General Perna as the chief operating officer. And then finally, the funding was $6.5 billion for the countermeasure development through BARDA and $3 billion for NIH. I have a quick little timeline there on the right showing some of the COVID vaccine candidates that have been selected by OWS to date. And if we hit the next slide, we can see how those kind of break down. Okay, so cool. This is a very simple infographic of what we can expect to see. I put 2 in from China as well because China has been performing quite well in some of their clinical trials. So there's a killed vaccine coming out of China. We have not gone that traditional route. I'm not going to steal the thunder of some of the other speakers. I think they have more in depth that they can dive into. But the US has not gone that route. And a lot of times the killed vaccines, they just don't get the response that you necessarily need. But this was one of the first and early vaccines that came to light. One of the most recent additions to the OWS portfolio has been Novavax, which has a recombinant subunit vaccine of the spike protein. We know that to be incredibly immunogenic. So that is a possibility. We have the new DNA or RNA vaccines. These 2 that I've listed here out of the OWS portfolio are both RNA. They're about 100 milligrams, I believe, in 2 doses. And the reason that I mention this is that, well, this is a new technology and it hasn't been scaled up in the manufacturing world before, which could put us at a disadvantage. I would like to also interject that Imperial College has a really interesting vaccine, which is a self-amplifying messenger RNA. And that's something that's going to stick within your system for 1 to 3 weeks. And it looks a little bit more like a virus and behaves a little bit more so that it attracts your immune system. So kind of more of a live bait scenario. And that's exactly why we have so many going after these vectored vaccines. These are vaccines that have been engineered so that they have components of both SARS-CoV-2, the virus that causes COVID-19, and also components of other viruses so that it looks and behaves more like a virus to our immune system. And here we have Oxford and AstraZeneca with their chimp adenovirus. We have Merck, and they are going after both VSV of the vesicular stomatitis virus as well as a measles form. We have the Jensen, which is also really interesting because they're looking at a single dose vaccine, and they're going to be comparing that in their upcoming clinical trials. And that's an adeno 26. And then we have Vaxart, which is an oral vaccine. Again, we haven't seen much on the way of data yet from Vaxart, but that should be interesting. And then finally, we have CanSino from China, which is going after adeno 5. So every single one of these offer different advantages and disadvantages, but I'm going to pause myself there to make sure that I give ample runway for Dr. Berry and AJ so that they can tell you more about it in their slides.
Well, thank you very much, Dr. Quattrochi. And Dr. Halamka, would you like to introduce our colleagues from Inference?
Oh, absolutely. So we have 2 esteemed colleagues joining us. From AJ is the head of biomedical research at Inference, which is a data-driven NLP machine learning firm in Kendall Square. He was formerly at Novartis. He did his postdoc at Stanford and his Ph.D. at the University of Cambridge. And then we have Dr. Michael Berry, who is a professor of medicine at Mayo Clinic. He's in the departments of infectious disease, immunology, and molecular medicine. He did his PhD at Dartmouth, just a few miles from where I am, Michael. I hope you climbed Mount Musalak a good number of times. And he works in gene therapy and vaccines. So welcome. And Brian, did you want to start with AJ, then go to Michael, or Michael, then go to AJ?
I think we had AJ first and then Michael next.
Okay, so AJ, you've just heard the comments of Lauren. Certainly welcome your comments as to where we are with vaccines and your experience, what the journey ahead is going to look like.
You might be on mute, AJ.
Yeah, thanks a lot for that kind introduction, John. Yeah, I'm very excited to be here and participate in this discussion. Let me quickly share my screen so that I can take you through some of the information that I put together. Right. Okay. So to start with, backing up a bit about nference itself, in addition to what John mentioned. So we are a biomedical data science company, and our mission is to make biomedical knowledge computable. And here we are taking different streams of approaches in triangulating insights based off of unstructured and structured data sets. And we're working closely with pharma partners and academic medical center partner, particularly the Mayo Clinic. And in this spirit of working with information where you use natural language processing methods to extract insights from literature or molecular data, things like RNA sequencing, protein structures, imaging, etc. And then at the other end of the spectrum, looking at the real world evidence itself from, say, patient notes or lab tests. So we are tying insights from all these different streams to help address specific needs in the context of diseases and healthcare. And in the realm of COVID-19 itself, nference has been involved in a bunch of different efforts along the different streams that I mentioned. So this includes analyzing information from literature, molecular data, and the real world data. And I invite you to check out some of the publications that are coming out. Moving ahead with the context of vaccine development itself in COVID-19, building up on what Lauren discussed, we have about 165 plus vaccine candidates, more than 25 of which are already in human trial and 6 in phase 3. There's a really neat infographic / tracker that the New York Times has put together, and that's what I'm displaying on this slide. And it's a great tool to check it out in conjunction with what WHO has put together as well. And here, this is highlighting where the different vaccines stand, with one particular exception, which is for expedited and emergency use, which has been left out. But this is the overall picture. But just looking into the specific types of vaccines itself, so you have them come in many different flavors, including the ones from Moderna, and where you're taking advantage of the genetic information from the coronavirus itself, SARS-CoV-2, and package it in a lipid biomolecule and deliver it to provoke an immune response. And like what Lauren mentioned, there's also a vaccine candidate coming up from Inovio, which I believe is a DNA-based vaccine. And the other flavors include things like a viral vector vaccine and the adenovirus-based vaccine candidates from Oxford, AstraZeneca, and J&J belong to this category. And then, of course, you have protein-based vaccines where you're challenging the immune system directly with components of protein components of the virus. And there are multiple efforts underway on this front as well. A couple of examples, including one from the Baylor College of Medicine and University of Pittsburgh. You also have whole virus vaccines, where the idea is to inactivate versions of SARS-CoV-2 itself, and that's the one that's under development from Sinopharm. And but definitely love to hear thoughts from Patty as well on the different versions of whole virus vaccines. And there's one other one which includes repurposing like existing vaccines, such as say the BCG vaccines for any potential cross protective effects. And there's at least one trial from Australia that is looking into this aspect. So at nference itself, we've also been looking at past immunization records of patients to see if there are any differences in the SARS-CoV-2 infection rates depending on the kind of vaccines that were administered in the recent past. And we have some preliminary insights that we are following up on. Another really exciting thing in terms of the vaccines that are being developed itself is that the information is also being shared with the public domain, if not in real time, like as real time as a peer review process would allow you to have a look at this. And you have a bunch of preclinical and clinical papers that are coming out based off of the different vaccines that I just briefly outlined in one of my previous slides. And here I'd like to shine some light on a couple of papers that are talking about the clinical data itself. So one is the mRNA-based vaccine from Moderna, and the second one is the one based from Oxford and AstraZeneca, which is your adenovirus-based vaccine. So two key components being safety and immunogenicity. In terms of safety for Moderna, it is like a phase 1 trial. It's open label with 45 healthy individuals between the age of 18 to 55. And these are two vaccinations, 28 days apart. And the 45 healthy adults, the total group was split into 3 categories, depending on the kind of dose that they were given: 25 micrograms, 100 micrograms, and 250 micrograms. And following up on the safety data, where you're essentially looking at the different symptoms, mild, moderate, or severe, both at the local level and at the systemic level. There were some amounts of mild to moderate symptoms that were seen upon the first vaccination, and specifically systemic adverse events were more common after the second vaccination in the higher dose. But the good news here is that none of these symptoms were trial limiting, and the dosage was from this particular study was also useful in deciding what goes into the phase 3, where I believe they're looking at the 100 microgram dose. And in terms of immunogenicity itself, in the publication that included antibody response using different kinds of assays, both neutralizing assays, and when you are also looking at specifically ELISA-based readouts. But in the interest of brevity, I'm just limiting it to one particular data set over here, where they are on the x-axis, you have the different dose groups and the time starting from day 1 to 57, and on the y-axis, you have the antibody response itself. What's observed is that there is seroconversion that's happening by day 15 in virtually all participants, and the the median magnitude of response in the 100 microgram and the 250 microgram categories is comparable to the median magnitude of response that you see from the convalescent donors samples. So that basically summarizes the Moderna and NIAID vaccine candidate, the mRNA-based one. And moving on to the University of Oxford AstraZeneca one. So this one was a phase 1/2 trial, which was single-blind and randomized control over 1,000 healthy adults between the age group of 18 to 55 years, and 5 into 10 part 10 viral particles were administered through injection. And looking at the safety data here, there were some local and systemic reactions, and there were two. There was a one-to-one matching done in this trial itself. So for one arm, it was the chimpanzee adenovirus-based vaccine, and the other one is like a meningococcal vaccine that was given, and the reactions that were seen were higher in the one with the vaccine candidate for COVID. But there was, again, the good news is that there were no serious adverse events related to the vaccine candidate itself. And in terms of immunogenicity, again, we're looking from at an ELISA-based response. So the y-axis is the antibody response, and the x-axis is the time in the different categories. So this is your control. And if we on comparison, we see that again around day 14, you have antibodies that are being generated, and this peaks at around day 28 and remains elevated till day 56, which was the data that's included as in this study. And the median levels again is comparable to what you see in the convalescent plasma samples itself. So just taking a step back, so we have 165 plus candidates, 25 of which are in human trials, and I suspect that a lot more data is going to be coming in the next few weeks, and these are like very interesting times. And I'd like to conclude this part by saying that we remain cautiously optimistic, quoting what's been said.
Great.
Yeah, with that, I'll conclude and pass it on. Yeah.
Thank you, AJ. And so before we get to questions, I wanted to have Dr. Berry please offer your introductory remarks on this as well. You might be on mute, Dr. Berry.
Yeah, sorry. Trying to find my appropriate desktop to share with you. So can you see that?
Yep.
Yeah, so Lauren and AJ gave great background. So I'm coming from a different background. I've worked on vaccines for about 28 years, and I've worked with a lot of the different ones that you've heard about. So I'm just going to try to skip a stone across some of the things that were said and maybe hopefully add some information. So sort of, most of you know a vaccine, the intent is to give you protection without actually having to suffer the disease. And in the vaccine world, the things we really want are antibodies, and ideally to neutralize COVID, SARS, before any infection occurs. So those are premier. And then T-cell responses are a response that is very valuable and you must have in order to actually get antibody responses. But these can actually be very important to kill infected cells after infection has occurred. But also the T-cell responses are probably the types of responses that might protect someone if we have, a SARS-3 that comes along. If there's enough similarity between subsequent coronaviruses, it's probably the T-cell responses that might help us out in the future. And sort of describe that. And so Lauren talked about the different types of vaccines. I just want to say that, when you guys get vaccinated, by far the most potent vaccines we have are what are called live attenuated whole pathogen vaccines. So if you think about measles virus or those types of vaccines, these are great because, they go in, they infect cells, they make their proteins, they generate antibodies and these important T-cell responses. And in some cases, you could be vaccinated once or twice and might have nearly lifelong immunity. But the problem is that if you, say, made a live attenuated version of SARS, there may be some risk that you might actually regenerate SARS itself. And so an example of that being a problem is that there's actually an outbreak of poliovirus that was actually caused by the attenuated vaccine. So we're not really seeing a lot of activity in terms of attenuated SARS virus as a vaccine, but if we could, that would be a very potent vaccine. And so you heard about the chemically inactivated vaccines. These are usually good at giving antibodies. They're usually not very good at giving T-cell responses. And so you need to use things called adjuvants. And it's these adjuvants that can, in many cases, give some of the reactogenicity of the vaccines. So you can also take the proteins out of the pathogen and make them, and these kind of work like the chemically inactivated vaccines. You can make peptides from the vaccines, and these probably won't make you antibodies, but they'll generate those T-cell responses. And then the last one, which we heard about a few times, is these so-called genetic vaccines. And basically, you're taking the genetic material out of whatever pathogen, but instead of taking all the genes that the pathogen has, you're just taking one or a few genes. And most of the vaccines we hear about are taking the surface protein, the spike protein from SARS, and putting that gene into other things like mRNA or plasmids or actually other viruses. And so these have they mimic the live attenuated vaccine, but you have no risk of causing SARS because you've removed just one or a few genes. And so, so if we talk about Moderna or Inovio, these are naked RNA or DNA vaccines. So these, have not evolved to get into cells efficiently. So they don't behave like a virus. So you tend to have to use things like chemicals to get these into cells so those genes can be expressed to make the proteins to activate the immune system. Or you can use a physical method like something called electroporation, where you actually inject the vaccine and then you give it an electrical zap to help it get into cells. And so the other solution, which we've heard, is to use viruses to deliver vaccine genes. And Mayo, we use viruses for a lot of things like killing cancer and whatnot. And so the ones you've heard about mostly are adenoviruses, Oxford, Janssen, Johnson & Johnson. And then you also heard about a vesicular stomatitis virus, VSV, from Merck. And, in terms of if you're thinking about these different genetic vaccines, in the field of vaccines, we kind of know the viral vectors tend to be more potent because they're basically stealing technology from evolution to get your gene into the cells to get it expressed. But they're also viruses, so they have biohazard. And the non-viral vectors are clearly the fastest to produce. You just synthesize DNA, you make it. So that's why we see Moderna and other DNA vaccines getting in faster. But they're generally weaker, and you may need to use adjuvants or delivery devices to do this. And so the other aspect, which maybe wasn't emphasized, is that, if we use viruses as vaccines, that they behave differently. So we hear about Oxford and whatnot that these are, a gentle version of the virus, and they are indeed. And but I mean, to kind of make that more clear, if we have a live attenuated SARS or we have a replication competent vesicular stomatitis vaccine, that virus will go into a cell and it will copy its DNA or RNA up to 10,000 times and make massively more protein than some of these other platforms. Problem is they create infectious viruses, and so, or can. And so if they're targeting SARS's ACE2 receptor, they actually could potentially cause some side effects that may mimic what we see with SARS itself. On the other end, we have the inactivated proteins, and we already talked about some of the issues with those. And then we have the Oxford, the adenovirus, the DNA, RNA, and so these are more potent, but when they enter a cell, they deliver their gene or their mRNA, and it stops. And so they don't make as much protein as, say, a live virus might do in terms of driving an immune response. And this may be very important if we, particularly in aging people whose immune systems aren't quite as robust as they could be. So this may be important. And then, some of the, and I think Lauren mentioned a type of vaccine that amplifies, that's kind of like an attenuated, but it will also do the amplification. That's I think she was mentioning an RNA vaccine from England, and so there are others, like Mayo has one that these are somewhere in between them that may be able to amplify things and sort of drive better immune responses. And so we already went over this data, so I won't belabor that. This is kind of encapsulated in the other talks. So the question is, what's going to be most potent? And as Lauren said, the ones we're moving forward with are really the fastest ones, and they may be adequate, but they may not be actually the most potent ones. So it'll be very interesting to see which ones come along afterwards, and in terms of which can maybe go in and be immunized once and provide sort of maybe long-lasting protection. So I'll stop there.
Great. Well, thank you, Dr. Berry, and thank you, fellow panelists, for that overview and your introductory remarks. I wanted to start the conversation off with the panel with this question, and Dr. Berry, I'll ask you to go first. With all of the news with Operation Warp Speed, and as you and AJ and Lauren have correctly identified, many vaccines in different stages of development, and people racing to get them out as fast as possible. One of the questions that we've been asked many times is, given all the advents and innovation that we have, why does it still take so much time for the testing of vaccines' efficacy and safety? And what, Dr. Berry, would be some thoughts on how to accelerate that, given the technologies that we have that we can bring to bear today?
Well, I mean, Lauren can probably address this kind of at the larger scale. In our case, we're more of a mom and pop kind of thing, where we built something and we're kind of having to bootstrap it ourselves. So whereas, if you're Moderna or you're Oxford, you've already got the system in place and you can kind of run with it pretty quickly. But, as was highlighted with Lauren, the top to-do thing is safety. And so that's what slows it down, I think, because you need to go through FDA or whatever the regulatory agency in a given country and satisfy them that this is going to be safe. And so you have to make a lot of vaccines. Traditionally, you have to make a human-like dose of vaccine tested in animals for safety, and that takes months, assuming you can make the sort of GMP-like vaccine in the first place. But I'll leave it to Lauren, since she's way more knowledgeable about that than I am.
Lauren.
Don't be silly. I think that you touched on a lot of the important points. Yeah, so the FDA or whatever your regulatory agency, that's going to be a lot of, we'll just say it, it's paperwork at some point that you have to generate. There are requirements that you have to meet to make sure that this vaccine is proven that it's both safe and effective. You mentioned GMP as well, or good manufacturing practices. That does not happen overnight. That's another thing where it's going to take time to actually ramp up to the amount that we're going to need. I mean, we're looking at 300 million vaccines, and those aren't just the vaccines, those are possibly 2 courses, possibly more. And we're not just looking for the US too, we're looking to expand internationally so that we can help other countries as well. This is not a problem just here, but everywhere. And it goes back to the World Health Organization One Health philosophy that we need to fix it everywhere if we're going to fix it here. Yep, scaling up to that. But I know that a lot of companies are now going at risk. They're manufacturing and scaling up, even though they don't even have full safety and efficacy profiles just yet. And so that's a huge step and risk for those companies to take on. The government is trying to buffer some of that with additional funding. But then it comes down to clinical trials. Clinical trials take time, setting them up, recruiting your volunteers, ensuring that you're getting and measuring the correct responses as your primary and secondary outcomes. Those all take time. I mean, it takes us for antibodies to develop, at least what we've seen is about 1 to 3 weeks. So that's a waiting period where we're checking again and again, coming back, seeing whether those antibodies have been produced. Are they sustained? I mean, now we have to have a longevity, like look over longer term, whether that's actually going to remain an effective technique to stimulate your immune system if it sees the virus again. So most of these clinical trials are extending out a year or more, but we're probably going to end up releasing the data and the vaccine before we maybe even get to some of those endpoints.
And so AJ, I might ask from your perspective at Inference, if that comes to pass, what Lauren and Dr. Berry are describing as we race to develop these vaccines and release the information, and there's a rush to have them shared in an equitable manner, and there are manufacturers already producing vast quantities of these, what would be some strategies that you would offer to mitigate against some of that risk as we go on this race in Operation Warp Speed?
Right. So taking a step back itself, the key bottleneck here is the clinical trial itself. I mean, there's no way we can accelerate biology to see if something is safe or not, right? So it's great that some of these steps are being taken in putting these different candidates up and testing them. But as what the previous panelists mentioned, the challenge is going to be in getting past the issue with the regulatory affairs and also the trial endpoints themselves, which, like as rightly pointed out, 1 to 2 years or even longer for a lot of these vaccines. Now, moving on to the second point that you raised about what strategies that we could think about, even in the event of scaling things up, is firstly identifying, are there any risk factors a priori that we can screen based off of the information that we already have about the different patients, like who are likely to be at higher risk of contracting severe disease compared to say individuals that have like mild or moderate disease. And this could be like a very effective data science problem as we consolidate data from different registries and academic medical centers. So if you frame that into that kind of a problem, that you have like a strong edge in getting a sense of which populations are at risk.
Got it. Thank you, Dr. Venkatakrishnan. Dr. Berry, one of the questions that I'm often asked is, these vaccines, as they make their way through phase 3 and ultimately perhaps get an EUA and are approved, where do you think we'll stand with regards to the efficacy of them? Measles and other vaccines that take years to develop oftentimes are touted as having, greater than 90% efficacy. What are your thoughts on where we'll land with any of these vaccines? And then a follow-up question to that would be, would it make sense for individuals to think about getting 2 or more vaccines if the efficacy for any given one is only 30 or 40%? You might be on mute, Dr. Berry.
Considering how many of these I do, you would think I wouldn't forget to do that. So actually in the vaccine world, a lot of times we, particularly when we're trying to go up against bad, hard bugs, we'll do what are called prime boost strategies. And so that might combine DNA and adenovirus or, those types of things. So I actually think that would be a great thing because it would also address some of the, there are these concerns that there aren't going to be enough people to volunteer or to be tested for vaccines. So it could be that if you failed, one of the other vaccines, you could then become, come in and, a lot of times if you get vaccinated and you don't really have a strong response, you've actually primed your immune system. So it's waiting just for a little stronger signal. So that is possible.
Great. And let's move on to one of the questions here in the chat window. Lauren, I'm going to come back to you with this one. Amber Bickford Cox asks, do you have any effectiveness concerns about each of the vaccines using novel, untested platforms it appears that the vaccines may be safe at this point, but the question is one of protection. And if I might add, the long-term potential side effects or safety or lack of protection associated with any given vaccine. If we're developing these in a year and then giving them out, we obviously don't have any safety data beyond a year. What are your thoughts on that?
Okay, so that was a pretty loaded question, right? So breaking it down, let me see if I can interpret some of this. So effectiveness concerns about each of the vaccines using untested platforms. Well, the only untested ones, right, there are the RNA and the DNA, the genomic, but we've done it in animals before and we've had success there. So again, this is already showing in clinical trials that these are working. So I'm not terribly concerned about effectiveness there. I should delineate that, you know, when you think about things, efficacy, you usually think about in terms of the individual patient. Effectiveness, you think about in terms of the wider population. So I would say, as far as if the intention is to talk about the effectiveness, so within the wider population, it might be that only time can tell. We are trying to diversify our clinical trials as much as possible. That's actually an ongoing concern, that the American population tends to be quite diverse. And so we need to make sure that our clinical trials are representative of that. We haven't done a terribly good job of that in the past. And I think that's something that's coming to light more and more. And that if we can make our clinical trials accurately reflect the population, then we'll be in a better place to predict what the effectiveness will be longer term. Otherwise, I would say the other platforms, we've used them all before. So especially looking at things like the vectored vaccines, we used those in Ebola and we found really good results there as well. And that's probably why Merck is again chasing a vectored vaccine. So it appears that the vaccines may be safe at this point, but the question is protection. In terms of sustained protection, like I said, we're probably going to need dosing more frequently. If it turns out to be true that our antibodies do end up waning and it's not like a MERS or SARS-CoV-1 scenario, which lasts for years, and if we do actually have to have our immune system boosted again, then we will end up implementing those routine vaccines. I will tell you that as far as safety concerns, and this is what I tell my aunts when they ask, is it's always better to get the vaccine than to get the legit infection. You know, 9 times out of 10, you don't think so, Dr. Very? No? Okay, he does. But yeah, it's never, the vaccines are there for a reason, and it's because you don't want the real thing. So I always advocate on behalf of the vaccine, even if you're going to have a reaction in terms of swelling or some mild cold symptoms, that's always going to be better than getting a legitimate infection.
Great. And so, AJ, I'm going to give this next question to you. You're a big data guy, and I'll preface it with, there are a lot of strains of you know, coronaviruses, right, and flu viruses, and any one vaccine can have a different efficacy against any one of those strains, and there's a certain level of personalization also based on the humoral or cellular immune response for any given vaccine for any one individual. And so Ben Zhao is asking, is there any data comparing the vast number of vaccines that we've been talking about and their suitability to different populations, be populations with different genetic variances, ethnic groups, ages? What are your thoughts on that? I guess if the data isn't there, do you think that given the number of vaccines that we have in trial, that there might be a scenario where certain ethnic groups would have a better likelihood of having a strong immune response to one vaccine over another ethnic group with another vaccine? Or is that more strain-based and then therefore something that we should be thinking about from year to year, different strains?
That's a really good question, right? And as far as the information itself is concerned around the strain, so there's centralized resources that are cataloging the different genomes of within SARS-CoV-2 itself. There are tens of thousands of publicly available genomes of SARS-CoV-2, both on NCBI, and there's another resource called GISAID, where you have like more than 50,000 different genomes that are sequenced and available. Now, in terms of the different vaccines themselves, so the question is like twofold, right? So we discussed the types of vaccines in terms of whether it's like genetic vaccine or whether it's like a virus-based vaccine and so on, or protein-based vaccine and so on. Now, one of the key components here is actually which component of the virus that you're using. So even if it's say just the spike protein, one can perform like a comparative analysis of this particular protein across the different strains and see how evolutionarily conserved they are or they're not. At a high level, they are not extremely diverse between the different viral strains, right? So it's not a case where there's in one strain is 50% identical to another strain. That's really not the case. Although there are some particular mutations that are being selected for as the pandemic itself is like progressing, right? Now, whether you would have like specific strain-based variants of these spike proteins that you would end up using in different vaccines, I think that should start off in the animal models first, right? And see that you're seeing some differences, and even before thinking about whether that makes sense to start having for different ethnicities or populations, you want to start off in the animal models. And if there are like significant differences that one can gauge, then it makes sense to think about translating it to humans. But even then, I think it's going to be a bigger logistical challenge now to identify the right match between the strain and the population / ethnicities of choice to have like these personalized or like tailor-made vaccines for specific subpopulations or populations.
Got it. Thanks, AJ. And I see we have Dr. James Lawler, an invited guest here, who is a regular contributor to some of our calls. Dr. Lawler, would you like to have your thoughts, sir?
Yeah, I just wanted to throw out a question for the group, given that there's been at least some suggestion of reinfection with divergent strains of the virus. What are thoughts and concerns about antibody-dependent enhancement, and how are we going to detect those types of events in clinical trials, which probably are not large enough to say with statistical significance whether that's happening?
Dr. Berry, would you like to take a stab at that one?
Well, as Lauren said, the plans are to track these vaccinees for quite long periods of time. I mean, the other thing that occurs to me is the use of the convalescent plasma, in terms of predicting, increased infections and whatnot. But I think that's still an open question. In animal models, antibody enhancement doesn't seem to be, that big of a deal. I think as long as you get, robust protection, it may be less of a problem, but I defer to maybe Lauren has some input on that.
So I guess following up on what you've been saying, and maybe I'm going a little off the rails here, but my sense is that the convalescent plasma as well as the therapeutics are going to be really key in our fight against COVID-19. So the vaccine is not going to be a magic bullet. It's not going to fix everything. There isn't going to be some massive, herd immunity appear all of a sudden. And because there are going to be people with different sensitivities to how effective the vaccine will be for them, I think really focusing in on some of the therapeutics and treating when it hits is going to be really key, as well as Brian works on. I know that you do a lot of work with the convalescent plasma, and that is, I think, going to be really important as we move forward, because the vaccines just aren't going to be able to cover all our bases, so to speak.
Got it. I wanted to close with one question, for everyone on the panel, and it's, if you had to choose, which one or two vaccines would you choose? Given, obviously, we're still in the beginning stages of this, but if you had to pick a winner or two, which ones would you sign yourself up for? And let's start with AJ, because you're smiling the most.
Well, like you said, Brian, I'm a data-driven person, so I would really defer to the data itself, right? So it's still like early days in terms of like the safety, and we have to start looking at the effectiveness data itself and efficacy data. So even for the two trials where we have some preliminary information, it's not really comparable in the sense that you have one way of single-blinded, the other one is open label, 45 patients, 1,000+ patients. So it's going to be hard to make like a decision at this point. But on a more lighter and philosophical note, like, why pick one winner? Let's hope there are multiple winners.
Fair enough. Lauren.
So I work with Inovio and their DNA vaccine. So I know that that's the one that I should choose. And if you guys, if you don't tell them, so I guess for the reasons that Dr. Berry laid out, I would say that I would lean toward either one of the vectored vaccines, or I would lean toward the self-amplifying RNA out of Imperial College. Having talked with their lead scientist, Dr. Shattuck, it sounds like a pretty promising technology, and definitely something that may help improve both the performance of the vaccine and the manufacturing speed if we can do it in just the genome, as well as make it behave a little bit more like a virus to our immune system. So I guess it would be a toss-up between that. And if I had to narrow down as far as vectored, I think we'll probably see a lot of promise coming out of the Johnson & Johnson vaccine, just because it's it could be a single dose, and we won't have to worry as much about compliance and people coming back to get their booster shot if it turns out that that works. But that said, yeah, those are my best guesses.
Very good. Dr. Berry.
Well, I have a dog in this show, so I'll, you can assume I think that's best. But I think you might have guidance from the 2014 Ebola outbreak. So the two vaccines that went into field studies, one was an adenovirus and one was a vesicular stomatitis virus. So, but each had, the more the virus is live, the more side effects you have. So I'll leave it at that.
Very good. Well, I know that we're 2 minutes over the hour. I'll put in my plug for Dr. Baruch and his vaccine out of Johnson & Johnson at Beth Israel. That's the one that I think the most promise is there. Well, I want to wrap this up and thank our panelists for joining us for this conversation. We'll be back next week with Dr. Campion leading us in another conversation around contact tracing. So thank you again for tuning in, and we'll see everyone next week. Have a great rest of your week, everyone. Bye-bye.